I'll start by saying a huge thank you to all those who have contributed to the research over the past 13 years. Like many groups, Sea Search and the Namibian Dolphin Project work off really small budgets, so each funder, collaborator, student, volunteer and data contributor is massively appreciated. I probably won't get invited to do many plenary talks in my lifetime, so I want to start off by dedicating this to an inspirational woman whose face and voice popped up throughout the preparation of this presentation, just like here in this clip.
Why is the blowhole off to one side?
This is a sperm whale.
Oh, amazing, I didn't know that.
Susan Rue wasn't an academic scientist, but she was an avid lover of nature, and for me and many others who knew her, she was a prominent voice of conservation in Namibia. She was never afraid to ask the questions many others were thinking. She inspired young Namibians to take up birding, leading groups during the biannual bird counts of the Walfish Bay Lagoon and Salt Pans, which is a Ramsar site. She put her hand up in local stakeholder meetings, which notably led to the first long-term underwater acoustic monitoring of coastal construction in Walfish Bay, Namibia. And as the role of citizen scientists becomes more important in conservation science, I want to dedicate this talk to Sue for her insurmountable enthusiasm, her passion and drive, and the support she showed to Simon and myself through our work as the Namibian Dolphin Project. As this is the crossover session, this talk will largely focus on the link between animal acoustic behaviour, acoustic monitoring, and the role of bioacoustic science in conservation. In Africa, many species and habitats suffer from direct conservation challenges or data deficiency, while generating new information is often hindered by small budgets and working in remote locations. Bioacoustics is an increasingly important tool in conservation science, but often the links between purely ethological and applied science are difficult to see, and therefore it sometimes feels a little difficult to justify behavioural research. There are many potential conservation applications of bioacoustics research as reviewed here, and my work really focuses mostly on the two first aspects highlighted in bold, so estimation of animal abundance and density, and individual acoustic identification. For me, the key advantage of using acoustic methods in conservation really boiled down to passive acoustics being a non-invasive, cost-effective method to collect information, and this is particularly useful for elusive species, which in my case includes marine mammals, who spend the majority of their lives underwater and out of sight. But to use acoustic monitoring to understand species, populations, or individuals, we must first really understand animal behaviour. Understanding the basics like species repertoire, production rates, and call function are all really key in our endeavour to use acoustic methods in applied conservation. More complex questions of identity encoding, understanding encoding of emotion, and cultural transmission are also very relevant, but sometimes harder to elucidate and require more time and focused, well-designed research studies. Many species exhibit different acoustic behaviours over time and from location to location, and these differences may result from environmental factors, ecological differences, genetic variation, learned and cultural differences, for example. Generalising from one area to another or one species to another is ill-advised, though happens surprisingly often. So the best acoustic programmes are based on a good knowledge of the acoustic behaviour of the study species, generated in the location of interest, and from data collected across a range of behavioural contexts and group dynamics. And this concept has really been fundamental to my research approach. I really enjoy behavioural science, but feel an ethical obligation to link my research to species conservation, and particularly when working on a continent with such high biological diversity, where basic information on many species, such as abundance, density, and ranging, is often lacking. Over the past 13 years of researching in Africa, I've been lucky enough to work on 4 African countries with 11 different cetacean species, including some of the offshore species. And I recently started work on other marine and coastal species, including Cape fur seals and coastal and estuarine birds. But in the interest of time, the focus of this talk is work conducted by myself and students on heavy-sized dolphins and humpback whales. Heavy-sized dolphins are endemic to the Anguilla ecosystem and found from Cape Town to southern Angola. Heavy-sized dolphins look like a harbour porpoise, and it's long been assumed that they sound just like a harbour porpoise as well. Both produce relatively long, narrow-band, high-frequency clicks, which in the case of harbour porpoise have a central frequency around 130 kHz, and for heavy-sized dolphins around 125 kHz. But until recently, our information on heavy-sized dolphin acoustic behaviour was based on just two studies. One conducted in the 1970s, which used an inappropriate sampling rate of 10,000 Hz, which could never have adequately sampled the acoustic behaviour of this high-frequency species, and the other which was recorded in one location, in the southern part of their range, and over a limited recording context. So heavy-sized dolphins look just like a harbour porpoise, but they don't always act like a harbour porpoise.
Just stay still so the sound trap is still underwater.
12.
So is it correct to assume that their acoustic behaviour will be exactly the same? And if it isn't, what does this mean for acoustic monitoring? This became more important as in 2014 to 2016, we conducted the first systematic visual and acoustic line transect surveys in the Namibian Islands Marine Protected Area in association with the Ministry of Fisheries and Marine Resources. We'd hoped to analyse this data using PAMGuard with built-in porpoise click classification, which we figured could work well with the narrowband high-frequency clicks of heavy-sized dolphins. But pilot data we collected from 2009 to 2012 indicated that as well as narrowband high-frequency clicks, which are very porpoise-like, heavy-sized dolphins might be producing bursts of pulses with a lower frequency. So in 2016, in Shearwater Bay near Lüderitz, PhD student Morgan Martin set out to record heavy-sized dolphins from a kayak using high-frequency sound trap hydrophones. These were lowered 1.5 meters below the kayak and sampling at a rate of 576 kHz. And behavioural notes were recorded throughout with a dictaphone, and a land team of volunteers checked on the behaviour and the location of heavy-sized dolphins groups during the recordings, as well as looking out for any other dolphin species which might have been passing through the area. In these data, we did find the narrowband high-frequency echolocation click trains and buzzes, which were linked to attempted prey capture. And there's a prevailing theory that the evolution of these particular click types represents a form of conversion evolution between several odontocetes, including porpoise, Kogia, and Keplerhynchus, as a form of acoustic crypsis. And indeed, as demonstrated here, these click types are likely not within killer whale hearing range. However, we also confirmed a second click type which was lower in frequency and much more broadband. And these were often produced in burst pulses, the frequency of which does overlap with the hearing range of killer whales. So switching to lower frequency burst pulses increases beam width and the active space of the signal. And it appears that heavy-sized dolphins may be using these riskier signals which can potentially be overheard by predators such as killer whales, in order to communicate over larger distances. And indeed, during her PhD studies, Morgan observed her study species being attacked and consumed by killer whales in Namibia. And looking at the behavioural context of burst pulse production in more detail, we showed that they were most commonly produced when animals were socialising. And at the recording site in Lüderitz, There are really good feeding opportunities, and whale-fed animals are often seen in tight-knit social groups in the inshore waters, and they're jumping around and interacting closely with each other here. So it's from these very specific and social behavioural contexts, which were displayed more often at this site in Lüderitz and others we have worked at, that we were really able to study the burst pulse production in heavy-sized dolphins. Under different behavioural contexts, such as foraging, narrow band and high frequency echolocation clicks are often produced, and burst pulses are absent. So this study really underlies the importance of recording context when defining the behaviour of a study species. Perhaps more interesting though, and a topic for more detailed future study, is that these burst pulses were occasionally organised into stereotyped pattern sequences, which supports work in other odontocetes, most notably in sperm whales, but also northern right whale dolphin and dusky dolphin, that temporal patterns in click production may encode information such as individual or group identity. Coming back to acoustic monitoring though, although heavy-sized dolphin acoustic behaviour is more complicated than we once thought, what I hope you can see here is that in many cases the standard narrowband high-frequency clicks were produced in recordings with the broadband burst pulses. And in fact, these narrowband high-frequency clicks were far and away the most common clicks produced and the loudest. So the detection of these was valid in passive acoustic line transect surveys of heavy-sized dolphins in the Namibian island MPA. And once we'd adequately described these clicks from Morgan's detailed behavioural studies, we were more confident in using automated detection and classification of narrowband high-frequency clicks in the freely available software PalmGuard. We used the default porpoise classification settings and shown here in red where we've got a detection of a heavy-sized dolphin. But we did spend a chunk of time to manually verify these results with visual sightings data to ground truth the detections so we were confident in our results. And this was necessary because we had a high number of false positive detections due to electrical noise in our recordings. So this seems like a fairly long-winded process to arrive back to the point that heavy-sized dolphins look like and sound like harbour porpoise. But by delving deeper into the acoustic behaviour, we're not only more confident in our data generated from the acoustic monitoring of this fairly undescribed species in a new geographic area, but we were also open-minded to the possibility of different behaviour, which has generated really interesting results for heavy-sized dolphins, which is reframing what we thought about this species, but also about the acoustic behaviour of other species in the genus. In our surveys, acoustic detection was particularly useful for overnight monitoring and also for monitoring in the deeper offshore areas. And by combining the visual and acoustic data collected, Morgan's been able to generate abundance estimates for heavy-sized dolphins in the vicinity of the Namibian Island Marine Protected Area. So I'll refer you to see Morgan's speed talk, an associated paper which was recently published, for more information on the methods that she applied and for an answer on how many dolphins there were in the Namibian Island MPA. So shifting focus to the other end of the spectrum, both in terms of size and frequency of vocal behaviour, the next part of my talk focuses on our research on humpback whales around South Africa and Namibia. And unlike heavy-sized dolphins, humpback whales are relatively well studied and there are some well-accepted knowns. So humpback whales in the Southern Ocean are increasing, and this has been facilitated by the International Ban on Whaling. There are many ways that humans are currently impacting whales, but apparently none so bad as directly killing them. Humpback whales migrate annually from cold polar waters where they feed to low latitude warm water areas where they breed. In Southern Africa, there are two main stocks as identified by the International Whaling Commission, breeding stock B and breeding stock C, which are then subdivided into further substocks. Broadly speaking, B migrates along the west coast to low latitude breeding grounds in Angola and Gabon, and breeding stock C moves along the east coast and breeds in several regions including Madagascar and Mozambique. There are seasonal peaks in numbers of animals sighted along the migration routes, which coincide with the northward and southward migrations between June and November. Since his PhD research in the 2000s by Jaco Verense, we have known that humpback whales aggregate along the west coast of South Africa between October and December, indicating an interrupted southward migration. But these aggregations have become larger and more impressive over the last 10 years, and particularly since 2015, with feeding seen until February or March time. These aggregations may be indicative of population growth, good foraging opportunities, or cultural transmission of prey information, perhaps. And the whales here are feeding on euphorid shrimp. So as biologists, we are all familiar with swarms of bees, flocks of birds, pods of dolphins even. But this is a supergroup of humpback whales. Ken Finlay and colleagues have described supergroups of 20 or more individual humpback whales, estimated to be within 5 body lengths of their nearest neighbour. These groups are forming as we speak, and this aerial image was taken in November last year, when 600 whales or more were observed in 1 hour of aerial surveys. And this is what they look like in action. And as you can imagine, collecting data from humpback whales when they are feeding in these large supergroups can be quite intimidating. Okay, so we're seeing very active feeding behaviour along the west coast of South Africa from October through to February and March. And we think these are most likely to be breeding stock B animals. But we can't rule out the possibility that some of breeding stock C are actually coming and feeding in the west coast waters. And this can be shown here in the satellite tags, where you can see animals from the east coast moving along to the west coast, and again in the second satellite track shown here, where this animal went all the way up to Namibia before heading back south again. And this is a track collected by the Department of Environmental, Forestry and Fisheries, which was collected last year. So are feeding whales breeding stock B, breeding stock C, or a mix? And as well as adults feeding in large supergroups, we also started to see other unusual feeding behaviour in our humpback whales, such as yearlings actively feeding in the near coastal waters around Cape Town. In 2019, we had around 10 to 12 yearlings feeding in the coastal waters, and these animals were between 7 and 8 metres in length. Again, this photograph is by Steve Benjamin. And as you can see here, the animals were incredibly close to shore, and they were apparently resident in around a 20-kilometre stretch of False Bay over this winter period. So like all good marine biologists, we took photo identification pictures of the dorsal fin and tail flukes, we catalogued the animals, and we gave them names. And through the season, citizen scientists tracked where these animals were using land-based photography, and in late October, timed with the southward migration, they seemed to disappear. But to our delight, 2 of these youngsters were spotted again in the exact same location, literally the exact same locations this year, and so we saw both Thelma and Sally returning. So both the feeding along the west coast and the return of young humpback whales who are also feeding and occurring in the exact same locations is pretty unusual. And it feels like humpback whales around South Africa are really rewriting the rule book of what is known about the species in these waters. So what might passive acoustic monitoring tell us about humpback whales in South Africa? Well, if we understand the acoustic behaviour and how this might change between contexts, over time and over space, we could be able to get information on occurrence, seasonality, habitat use, density, and even stock structure, maybe providing insights into which stocks are feeding along the west coast in these large supergroups. So here, acoustic monitoring is one of the many tools in the box to understand humpback whales in South African waters. So in general, humpback whales are relatively well studied, and there is a large amount of information on their acoustic behaviour. We know that males produce complex pattern songs which are prolific at the breeding grounds. Males converge on the same song type in a location, and this song type changes slowly over time. And this was really well summarised by Ellen Garland in her talk earlier at this conference. Both sexes produce a repertoire of non-song calls, which can be quite diverse. We know less about the function of these calls, but research into non-song calls has increased rapidly over the last 10 years. In general, though, there is very little information on the behaviour of humpback whales moving through or occurring in South African waters, and little known on their acoustic behaviour. So we're addressing this from several perspectives, recording both at the feeding areas on the west coast of South Africa and also within False Bay. We've been conducting focal group follows while simultaneously deploying a dipping hydrophone. We've combined static acoustic monitoring using our moored sound trap recorders with remote observation from land-based platforms. We've analysed acoustic data collected from individual animals using CATS tags, which had been deployed by the Department of Environment, Forestry and Fisheries. And we've been looking at the feeding behaviour from underwater video. A large part of our data comes from boat-based data collection. We work on a fairly small budget, and we often use just a single hydrophone deployed from the research boat in coastal areas. Combining the data from the focal group follows collected in this way with data collected from the tags, which Catherine Adian talked about earlier, we've logged over 12,000 sounds, including both song and non-song calls. And from these, we've documented a repertoire consisting of more than 16 different call types, with visual classification of the most abundant 11 call types supported by classification and regression tree analysis. And the most common call types, such as the whoop and the grumble sounds, are structurally similar to those observed in other geographical areas. So animals feeding in these large supergroups are highly vocal. And with such high numbers of animals, it's not really surprising that there's a high production rate of sounds. So it's possible that we could use acoustic monitoring of the most clearly defined sounds, such as the grumble or the whoops, to identify when groups are present and feeding. But there's also prolific song recorded when groups are feeding, which, although really interesting, was a little surprising, but also muddies the picture and makes acoustic monitoring of feeding whales more difficult. And this occurrence of song also generates other interesting questions, such as whether song functions within the feeding context somehow, and whether cultural transmission of song might occur at this feeding area. And these are questions that we hope to address later this year and also into next year. In fact, the more we work on humpback whales, the more we detect humpback whale song. And although now widely accepted that humpback whales do sing on migration routes and at feeding grounds, the amount of song and the timing has surprised us a little. And perhaps this is because it hasn't been well documented in South African coastal areas before. So we detect song on the northward and the southward migration times, on the west coast and within False Bay. Around Cape Town, we've confirmed humpback whale presence through song recordings from June through to December. In our first analysis, based on acoustic detection at 4 hydrophones located in False Bay, we found that song transmitted across False Bay, and we detected more song and louder song on the most western hydrophone. We weren't sure where our singers were and whether they were perhaps south of Cape Point. In August this year, we were really thrilled to find active singers within False Bay, which were recorded for 2 hours, and it's this year's song which is playing now. But like this spectrogram of multiple simultaneous singers, our current understanding of humpback whales generated through acoustic methods is pretty confusing. Using acoustic monitoring to indicate occurrence of singers is pretty straightforward. But whales will be present and not singing. Song changes between years and between areas, making automated detection and classification more difficult. There's also a high amount of overlap between the sounds produced by humpback whales and say other species which occur in the same areas, like southern right whales, which also makes species classification difficult. Also, propagation distances differ with environmental conditions, so actually pinning down the location of your animals can also be pretty difficult. So we have a long way to go, and much bigger budgets really are needed before the more complex questions on humpback whales can be answered using acoustic monitoring. So moving on to the final section of this talk, and this is where it gets a little bit personal, so I do throw up a warning, because planning this talk has been quite a reflective process. And I want to dedicate the remaining time to sharing some lessons learned along the way from the past 13 years of working here in various countries in Africa. So there's the obvious ones like being nice to each other, staying polite, saying yes to opportunities when they arise, and of course attending conferences. Perhaps though, don't volunteer for plenary at a conference you're also organising. And another would be to be prepared. So know what data or research questions you're interested in, and then if the chance arises, being in the right place, at the right time, and with the right equipment can be really great. This may be easier said than done, but here we've recorded 2 rare offshore species at the V&A waterfront in Cape Town through being prepared and with a good team ready to go and the right equipment. At the top we have recordings of a dwarf sperm whale, which are an offshore species, and at the bottom a group of false killer whales. And the next is to find a really good mentor and then stick with them. So you can't really underestimate the importance of long-term relationships in science. And just to show you this, this is a photo of us at St Andrews University earlier in this year, in January. And here we've got Vincent Yannick, who is my PhD supervisor, who I first volunteered for in 2003, as well as Julie Oswald, Volker Decker and Doug Gillespie, who continue to be really helpful people to bounce ideas off and to chat to about acoustic monitoring. These academics are also now helping my PhD students in their studies, and in 2021 I'll co-supervise a student with Julie. So these long-term academic relationships are very, very useful and can be very productive. Which brings me to my next point, which is really to be prepared for the long game. Just a brief background of my career trajectory. I did an undergraduate in marine biology, which I graduated from in 2003, and went on to volunteer at St Andrews University on a signature whistle research project. In 2004, I applied for a research assistant position on humpback dolphins with the Leverhulme Trust, which I was close to getting, but unfortunately didn't get. And in 2006, I started a PhD at St Andrews University. As part of my PhD research, I was lucky enough to go to Zanzibar in 2008 to conduct research on the acoustic behaviour of bottlenose dolphins. And whilst in Zanzibar, I witnessed the dolphin bycatch issue firsthand. And dolphins that were caught overnight in nets would be bought for around $10 by the fisheries inspectors, so that scientific data such as the size, sex, species and things like that could be collected from the carcasses, before the carcasses were then distributed for consumption or used as bait. I'm sorry if these photos are a little bit graphic. And as well as my study species being caught in nets, humpback dolphins were also being caught in fishing nets. and the Indian Ocean humpback dolphins have subsequently been listed as endangered. And although I don't work on humpback dolphins in Zanzibar, my work in South Africa includes a strong conservation focus as part of the SUSA Consortium, which is a dedicated team of scientists working for the conservation of humpback dolphins. And I'm going to post these two infographics in on the conference site so you can get a little bit more information about bottlenose dolphins and humpback dolphins around Namibia and within South Africa. And this work on bottlenose dolphins and on humpback dolphins includes looking into whether we can use signature whistles, which are individually distinctive whistle types used by dolphins, in a mark recapture framework to understand the occurrence and density of threatened dolphin populations. And we've called this project the SWORD project, which stands for Signature Whistle Occurrence, Recapture and Density. And several members of our team will talk about this project in more detail within the conference. And we recently published the first results of this project in the Journal of Mammalogy. And here, the signature whistle abundance estimates were very similar to the abundance estimates generated for the same population using photo identification. So moving on there, the best advice I received some years back when interested in dolphin research, which apparently a lot of people are, was to diversify. And it's taken me a while to get there, but working with new species has resulted in very literal links between bioacoustic research and conservation. So for instance, by recently recording endangered Cape cormorants nesting on boats in Cape Town, and here we have a microphone strapped to a boat. We've also been close enough to see that the nests are actually built of plastics like cable ties and plastic bags and also fishing lines. And the problem here is that not only do the adult cormorants get caught in the fishing lines, but also that the newly fledged chicks also get caught in these lines and can die as a result. And as well as issues with the nest being built with plastics, we're also seeing nets resulting in direct mortality on the boats moored. So this is a very recent observation, but we're feeding our results back to the relevant authorities and boat users, which will hopefully result in direct changes in terms of how boat organisers deal with having nesting birds on their boats, and also how the nets are used on the boats and whether or not we can reduce the amounts of nets on boats, which also cause cormorant deaths. And it's a similar story from working with Cape fur seals. So Mathilde Martin and Isabelle Sharia have been talking at this conference about the work that we've been conducting on Cape fur seals in Namibia. And by working at the colonies, you get a chance to get very close and personal to the Cape fur seals. As you can see here with the animal just sniffing the microphone. And this is work that's being done by Mathilde Martin as part of her PhD on the vocal behaviour of Cape fur seals. But whilst working at the colonies, we've also started to document plastic entanglements on a large number of animals, which is perhaps less of a conservation issue than a welfare issue, but it definitely is something which we need to address, both for the animal welfare perspective and also as a reflection of our behaviour and our approach to plastic management. And by having a team of researchers interested in the acoustic behaviour of Cape fur seals has also meant we've had more scientific input into a recent abortion event which has taken place at the field site at the Pelican Point colony in central Namibia. And here between 5,000 and 7,000 pregnancies have been aborted in early October this year. And so having more people involved working on different aspects of the seal behaviour and ecology has meant we've been better placed to respond to this event. So these are just a few examples where focused behavioural observation, often necessary for bioacoustics research and data collection, have had direct relationships with conservation issues. And if this is all starting to look a little bit depressing, I apologise because it doesn't get much better in the next slide. Because I really recommend that you look after yourself and each other. And if this is all starting to look a little bit depressing, I apologise because it doesn't get much better in the next slide. Because I really recommend that you look after yourself and each other. And this brings me to my final point, which we must try and find a work-life balance. Find your support network and stick with them. And for me, this has been Simon Elwyn. We've been working together alongside for around 11 years. We run together the Namibian Dolphin Project and Sea Search, and Simon has contributed massively to all the research summarised here in some way. Which almost looks romantic until you see that we're mostly in the field or behind a computer. We conduct necropsies, and there's a whale head in the bag. And life looks a lot like this, which is a real juggle of work and kids and field work and conferences and grant writing and trying to do some science in between, publish some papers and organise a conference. And while it's been very busy, looking over the pictures to prepare for this plenary has been a real pleasure. And over the past 13 years, working in 4 different countries and conducting the bioacoustics research we have has been a great adventure. So with that, I'll thank you for your attention and I'll be available to take any questions.